Hi, good morning. My group mates described what recombinant DNA or genetic engineering is and discussed the power of genetic engineering. Now, let's move on to the discussion of the ethical and social issues regarding genetic engineering and biotechnology. On my report, I will be focusing on the release of genetically modified organisms, its biosafety and implications. So first, I will tackle about its potential ecological effects and then the regulating policies concerning it. So, the early concerns about the potential dangers associated with recombinant DNA technology focused on the possibility that hazardous new pathogens might be created. Like for instance, what do you think will happen if cancer cell genes were transferred into bacteria or viruses? Scary, isn't it? So, to guard against such microbes, scientists developed a set of guidelines that were adopted as formal government regulations wherein there is a set of strict laboratory procedures designed to protect researchers from engineered microbes and to prevent these microbes from accidentally leaving the laboratory. But today, most public is concerned not on the hazards of recombinant microbes, but on GMO, or genetically modified organisms. So, in common language, genetically modified organism, or GMO, is one that has acquired one or more genes by artificial means from another species or even from another variety of the same species. So next. Wow, enticing, isn't it? Some salmon, for example, have been genetically modified by addition of a more active salmon growth hormone gene. So in effect, these transgenic salmon grow larger and faster than the common salmon, and they mature faster too. By any chance, do you have any idea that the salmon you are eating or have eaten might be a genetically modified one? However, genetically modified organisms that contribute to our food supply are not animals, but crop plants. Genetically modified organisms are widespread in United States, Argentina, and Brazil. Together, these countries account for over 80% of the world's acreage devoted to such crops. In the United States, the majority of corn, soybeans, and canola are genetically modified crops. Now, one potential ecological effect is that transgenic plants might pass their new genes to close relatives in nearby wild areas. We know that lawn and crop grasses, for example, commonly exchange genes via pollen transfer. If crop plants carrying genes for resistance to herbicides, diseases, or insect pests, pollinated ones, the offspring might become superweeds that are very difficult to control. So, many strategies are pursued with the goal of preventing gene escape. For example, if male sterility is engineered into plants, these plants could still produce fruit and seeds if pollinated by nearby non-transgenic plants, but they would produce no viable pollen. The second approach involves genetically engineering apomixis, wherein the embryo and endosperm develop without fertilization. The transfer of these traits to transgenic crops would minimize the possibility of preventing transgene escape via pollen, because the plant could be male sterile without compromising the fruit and seed production. A third approach is to engineer the transgene into the chloroplast DNA of the crop. Chloroplast DNA in many plant species are inherited from the egg. So, transgenes in the chloroplast cannot be transferred by pollen. The fourth approach for preventing transgene escape is to genetically engineer flowers that develop normally but fail to open. Consequently, self-pollination would occur, but pollen would be unlikely to escape from the flower. Another potential ecological effect is that a transgene encoding a pesticide-like protein might cause plants to produce pollen toxic to butterflies, a possible effect on non-target organisms. However, scientists with the Agricultural Research Service concluded from a 2-year field study that butterflies are unlikely to be exposed to toxic level of pollen. As for the risks to human health from genetically modified foods, some people fear that the protein product of transgenes might lead to allergic reactions. Although there is some evidence that this could happen, advocates claim that these proteins could be tested in advance to avoid producing products that can cause allergic reactions. Today, government and regulating policies throughout the world are grappling on how to facilitate the use of biotechnology in industry, agriculture, and medicine while ensuring that the new products are safe. One regulating policy is to label genetically modified products. A study investigating voluntary labeling in South Africa found that 31% of products labeled as GMO-free had a GM content above 1.0%. In Canada and USA, labeling of GM food is voluntary, while in Europe, all food, including processed food, which contains greater than 0.9% of approved genetically modified organisms, must be labeled. There are differences in the regulation of GM crops between countries, with some of the most marked differences occurring between the USA and the Europe. Regulation varies in a given country depending on the intended use of the products of genetic engineering. In the United States, such applications of biotechnology are evaluated for potential risks by various regulatory agencies, including the FDA, the Environmental Protection Agency, the National Institutes of Health, and the Department of Agriculture. Ethical considerations as well as concerns about potential environmental and human health hazards will likely slow some application of biotechnology. But we see there is always a danger that too much regulation will stifle basic research and its potential benefits. Therefore, scientists and the public must assess the case-to-case basis on the potential benefits of transgenic crops versus the risks that the society is willing to take. We must base our discussions and decisions on the sound scientific information and rigorous testing rather than the reflexive fear and blind optimism. I am Danica from Untiveros, and this concludes my report. Thank you.